Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine the impact of a stroke. It's a leading cause of death, yes, but also a major cause of long-term disability, really worldwide.
Yeah, it's devastating.
And the immediate priority is always, restore blood flow to the brain fast.
Absolutely. Time is critical.
But even when that happens, the damage isn't necessarily over, right? The brain's own response involves inflammation. And while some of that's needed for cleanup, this secondary wave of damage from too much inflammation can happen hours, even days later.
And that causes real lasting challenges for recovery. And current standard treatments are very, very time sensitive. They mostly focus on that initial window, getting the blood flow back.
Right.
So there's a big need for therapies that can tackle this later phase, this secondary injury, and actively help the brain repair itself.
Absolutely. So what if a potential new path to helping stroke recovery came from somewhere totally unexpected, like drugs originally designed for cancer?
It sounds surprising, doesn't it? But that's exactly the angle explored in this recent study we're diving into today. It's from researchers at the National University of Singapore Medicine.
Okay.
They looked into using a class of drugs called Histone Deacetylase Inhibitors. Let's just stick with that full name. Histone Deacetylase Inhibitors to protect the brain and boost recovery after stroke.
And they were specifically looking at how these drugs affect the brain's own immune cells, the microglia.
Exactly, the microglia. We've been looking at a health news article about their work and also dug into the original scientific paper itself, published in Gilead.
So our mission here is to unpack how these drugs, known for working on cancer cells, might be repurposed for stroke recovery.
Yeah, we want to look at what the study actually found, how these drugs might work on those microglia, and importantly, what this could all mean for treating strokes down the line.
Yeah.
Maybe opening up new treatment windows.
Okay, let's jump in. What was the core idea? Why look at histone deacetylase inhibitors for stroke in the first place?
Well, these histone deacetylase inhibitors, they're a class of drugs already being used or looked at for different things, like cancer, yes, but also some neurodegenerative diseases.
Right.
The key thing is how they work. They influence gene expression. Basically, they can change which genes get turned on or off inside a cell, but without changing the actual DNA code itself. It's part of something called epigenetic regulation.
Okay, so they're like changing the instructions the cell follows without rewriting the instruction manual itself. And the idea was they could use this to affect brain cells after a stroke.
Precisely.
Oh.
And their specific target was the microglia. You can think of microglia as the brain's main immune defenders, the cleanup crew.
Got it.
After a stroke hits, they activate really quickly. And initially, that's good. They start clearing away damaged tissue and debris.
Makes sense.
But if they stay in this hyperactivated state, they can start releasing inflammatory stuff that, unfortunately, damages nearby healthy tissue. This happens in the hours and days after the initial event.
So they go from being helpful first responders to actually becoming part of the problem later on.
Exactly that. They can really contribute to that secondary wave of injury we mentioned. So the researchers thought maybe these histone deacetylase inhibitors could step in and sort of shift the microglia away from that harmful inflammatory state.
Towards a more protective healing state, like reprogramming them.
Yes, reprogramming is a good way to put it.
Yeah.
Could these drugs essentially change the microglia's job description on the fly?
That's a really fascinating concept. Turning the cell's own programming from damage to repair. How did they actually test this idea?
So they used a pretty standard lab model. They simulated an ischemic stroke that's the type caused by a blocked blood vessel in mice. This lets researchers control things and study the biology closely.
Okay, mouse model.
Yeah. And they used a specific histone deacetylase inhibitor called sodium butyrate. They gave it at 2 different times. One dose shortly before they induced the stroke, and another dose several hours after they restored the blood flow.
So looking at effects both before, maybe preconditioning, and also during that critical time after blood flow comes back, when that secondary injury kicks in.
Yes, hitting that key reperfusion phase. And to see what was happening really closely, they used some pretty sophisticated techniques. For example, special stains to actually see and measure the brain tissue damage.
Okay.
And they used advanced microscopy to look really closely at the shape of the microglia. Plus, techniques like spatial transcriptomics.
Spatial transcriptomics. Okay, break that down for us simply.
Right. Imagine you've got a map of the brain slice you're looking at. Spatial transcriptomics is like technology that lets you see, right on that map, which genes are switched on or off in specific cells in specific locations.
Wow.
So it gave them this incredibly detailed picture of the genetic instructions being followed by cells like the microglia in different brain areas hit by the stroke.
Okay, so they had the model, the drug, the timing, and these high-tech tools to see what was happening. What did they actually find? What was the difference between the treated mice and the untreated ones?
The results were pretty striking, actually. One of the biggest findings was a really dramatic reduction in the amount of brain tissue damage in the mice that got the drug.
How dramatic.
They reported a 60% reduction in the volume of the damaged tissue compared to the untreated stroke group.
60%. That's a huge protective effect.
It really is significant. And they could see this clearly with that staining technique. The damaged tissue shows up differently, so they could measure the stroke size accurately.
And this wasn't just something you could see under a microscope, right? Did it affect how the animals were actually doing?
Yes, exactly. The mice treated with the Histone Deacetylase Inhibitor, they showed better physical recovery. They performed better in simple behavioral tests that are designed to check neurological function after a stroke.
Like what kind of tests?
Things like how well they moved around or how they navigated simple tasks. It showed there were functional benefits that matched up with that reduced physical damage.
Okay, so less brain damage, better function. Now, back to those microglia, the main target. Did this study show the drug was actually changing them, their shape or behavior?
Yes, and this is where that reprogramming idea really comes into focus. In the untreated mice, the microglia in the injured areas changed shape. They became kind of rounded, swollen, lost their usual delicate branches. That's called the amoeboid shape, and it's often linked with aggressive inflammation. But in the mice that got the treatment, the microglia in those same affected areas looked remarkably different.
How so?
They mostly kept their normal shape, which is more ramified-like, a small cell body with long, thin arms reaching out.
Almost like they were staying in their normal surveillance mode instead of going into that sort of attack mode.
That's a good way to think about it.
Yeah.
Their shape suggested they weren't shifting into that aggressive inflammatory state. And the researchers didn't just eyeball this. They used detailed imaging and 3D reconstruction software to precisely measure these shape changes. It confirmed the treatment effect.
Okay, and you mentioned earlier that these drugs work epigenetically, affecting gene expression. Did they show that happening in the microglia?
They did. That's kind of the deeper mechanism they uncovered. The study looked at specific epigenetic markers. These are like little tags on the proteins the DNA wraps around. These tags influence whether genes are easy or hard to read and turn on.
Right.
They found that in microglia after a stroke, certain markers were changed in ways that usually promote inflammation. But when they gave the histone deacetylase inhibitor treatment, these markers went back closer to normal levels.
So the drug was directly tweaking those settings on the DNA packaging, essentially.
That's what the evidence points to, yes. And the spatial transcriptomics data backed this up. It showed a massive shift in the actual genes being expressed by the microglia in the treated animals.
What kind of shift?
Well, genes linked to neuroinflammation were significantly turned down. And importantly, genes involved in helpful processes like neuroprotection and phagocytosis.
Phagocytosis, that's
Yeah.
the cell eating debris.
Exactly, the cleanup process. Those beneficial genes were boosted or kind of rescued back towards healthy levels.
Interesting. So the drug didn't just put the brakes on inflammation. It seemed to actually hit the gas on cleanup and protection pathways.
That's the really exciting part, yeah. It suggests it wasn't just stopping the bad stuff, but actively promoting the good stuff. A comprehensive shift in the microglia's function from harmful to helpful. And using that spatial transcriptomics, they confirmed this was happening right within the specific brain regions affected by the stroke, places like the hippocampus, thalamus, striatum, cortex, though the exact pattern of gene changes varied a bit by region, which just highlights how complex the brain's response is.
Okay, so we've got reduced overall damage, better function, microglia changing shape back towards normal, and these fundamental shifts in their gene activity promoting healing. What are the sort of bigger picture implications here?
Well, one important related finding was that they also saw protective changes in gene expression in other key brain cells nearby, specifically neurons and astrocytes, which are another type of support cell.
Ah, so it's not just the microglia.
It suggests that reprogramming the microglia has positive knock-on effects for the whole neighborhood of cells in the injured area. When the immune cells are behaving helpfully, it seems to support the neurons and other critical cells too.
It makes sense, like creating a better environment for the whole brain area to heal.
Exactly. But the major implication, I think, goes back to that potential treatment window we talked about earlier.
Right.
Because this approach targets that secondary wave of injury, the inflammation and damage unfolding over hours and days after the initial stroke event and after blood flow is restored. It could potentially offer a much longer window for treatment than many current therapies.
Which could be huge for patients who maybe don't get to the hospital within those first few crucial hours.
Potentially, yes. By dampening that later inflammation and actively promoting repair and debris clearance, these types of drugs might significantly improve long-term functional recovery, reducing disability. And the researchers themselves seem quite optimistic about this.
What did they emphasize as the key takeaway from their work?
Professor S. Thamim Dean, one of the lead researchers, talked about the tremendous potential for clinical translation. He specifically mentioned using these histone deacetylase inhibitors to dampen inflammation in the brain and promote recovery in stroke through microglia-centric mechanism.
Right, focusing on those microglia as the linchpin.
And another lead, Professor Thurima V. Arumugam, really highlighted how the drugs seem to precisely reprogram microglia in stroke-affected brain regions, transforming them from inflammatory agents to healing promoters. That functional transformation is the core idea.
And didn't Dr. Kevin Jairaj call it an out-of-the-box approach?
He did, yeah. He emphasized finding alternative targets like this represents an out-of-the-box approach for treating stroke and potentially other conditions too, where neuroinflammation is a big factor.
So could this microglia-focused strategy be relevant for other brain conditions?
It's certainly possible. Things like neurodegenerative diseases often involve chronic brain inflammation, so understanding how to modulate microglia could have broader applications.
That's a potentially wide impact. Now, okay, this all sounds very promising, but we should always talk about limitations, especially at this research stage. What are the important caveats here?
Absolutely crucial. First and foremost, we have to remember this was a study done in laboratory models, specifically mice.
Right.
And while animal models are essential tools for understanding biology and testing therapies, what works in mice doesn't always translate perfectly to humans. Human brains and human strokes are much more complex.
That's always the big hurdle, the translation to people. What else?
The other key thing is the time frame they focused on. This study primarily looked at events within 24 hours after the simulated stroke.
Okay.
So while they saw clear changes in repair pathways and observed better behavioral performance within their experimental setup during that time, they didn't conduct an extensive battery of long-term behavioral tests tracking recovery over weeks or months. That wasn't the main goal for this particular paper.
So their focus was really on nailing down the acute effects, the mechanism of action at that peak inflammation time.
Exactly. They wanted to deeply investigate that immediate inflammatory response and how the drug affected cell behavior and gene expression right then at that 24-hour mark, which is optimal for studying that specific phase.
So it gives us really strong evidence for the mechanism and the acute protective effect in this model, but the full story on long-term functional recovery would need more, longer studies.
Precisely. This study leaves really important groundwork, showing how it might work, and that it does work acutely in the model. It definitely paves the way for that necessary future work.
Okay, so pulling this all together then, this deep dive looked at research showing that by repurposing these drugs, Histone Deacetylase Inhibitors drugs looked at for cancer scientists showed, at least in a lab model, they can significantly protect the brain after a stroke.
Yeah, and the core mechanism seems to be changing the job description of the brain's immune cells, the microglia, shifting them away from promoting harmful inflammation and towards supporting helpful repair and cleanup.
And the potential here is a new strategy that isn't just about that initial emergency phase of restoring blood flow, but about intervening later during that secondary injury phase, possibly over a longer time window.
Right, aiming to actively reprogram brain cells like microglia, and maybe astrocytes too, to create an environment that fosters repair and helps restore neurological function over the longer term.
It leaves you thinking, doesn't it? Consider how exploring these really complex, sometimes surprising roles of cells like microglia, and looking for answers in totally different fields, like cancer research, how that could fundamentally shift how we tackle really devastating conditions like brain damage after stroke. What other hidden connections might be out there in science, just waiting for someone to connect the dots and offer new hope?
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.